Thank you to the Academy for the invitation to come here to visit a fantastic city of the world and also an incredible space to be in. I have to say I don't often get to speak in such spaces, so it's lovely to be here. In my career, I've spent a lot of time doing work with my colleagues around the basic biology of particular symbiosis of Wolbachia and insects. But in recent years, as I've gotten older and started to get more grey, I've become much more interested in the application of the basic work that we had done earlier. And so what I want to talk to you today is much more about the applied use of the Wolbachia endosymbiosis for controlling mosquito-transmitted diseases and the work that we are doing. We're working in a fairly large consortium of people at the moment, and we call the project that we work on Eliminate Dengue, our challenge. And we use it in a way, I guess, to talk about the aspiration of our work and where we want to go. But also, we put our challenge on the bottom because we appreciate the enormity of the task. And so if you were to look at dengue, which is a human virus transmitted by Aedes aegypti mosquitoes throughout the world, we see that around 40% of the global human population is at risk of getting dengue in any given year. The global government investment in trying to control dengue, and this is in many countries which are quite poor, amounts to around close to $9 billion every year. But on top of that government investment, there is around an additional 45% of the cost is borne by families, and often families in very poor countries. And that these costs for having to deal with dengue cases within their houses can be up to 3 times the family's monthly income. And so this disease has a devastating impact, not just on the direct health of individuals, but also on the social and economic costs, indirect costs. And certainly if you were to go to a country like Vietnam, for example, just to name one, during a major dengue outbreak, you would find the hospital system clogged with dengue patients in the corridors and the health system unable to deal with just the volume of people entering the system. And then the same mosquito we know in the last year or so is also responsible for transmitting a very similar flavivirus called Zika, and it has emerged and spread globally throughout the last 2 years or so to become a major problem. And what has been so alarming about this particular virus is the difficulty that it's causing in developing fetuses, where we see microcephaly being expressed, so babies being born without full development of their brain, and these small head sizes. And in Brazil, for example, since February 2016, there's been around 2,200 reported cases of microcephaly in babies, and that's against a normal background of around 160 cases nationwide. And Zika is spreading throughout the tropics of the world at the moment, up to 75 countries, and over 60% of those countries in the last 2 years. And so we're seeing these Aedes aegypti transmitted viruses emerging across the world at the moment. Just right now, at the moment, we have a problem with yellow fever, which we haven't seen major outbreaks of yellow fever for some time. And now we're seeing in West Africa and in South America significant numbers of yellow fever cases. And so we're seeing waves of these epidemics emerging from these Aedes aegypti transmitted viruses. So I want to talk about a way that we're working as a group of people to think about a different way of trying to control these viruses, because fundamentally the current methods of control don't work. If they did work, we wouldn't see what I just showed you on the last 2 slides. So we have a major problem where current control measures are not working, and what I would like to present to you is a different approach. And this approach uses the bacterial symbiont known as Wolbachia. It occurs in nature naturally in up to around 60% of all insect species. It's not exactly clear how many species of insects have Wolbachia, or actually how many species of insects exist at all. But we know that many insects naturally carry Wolbachia. And because of that, it's a relatively benign and safe agent in the environment. And the real key observation, which I'll show you some data on, is that when insects, or when mosquitoes particularly, when we introduce Wolbachia artificially into the mosquito, we find that the presence of Wolbachia provides protection against the replication of a number of human pathogens, including dengue, Zika, and chikungunya. So, Wolbachia is a really interesting microorganism. And one of the reasons that it's so interesting is that it doesn't spread infectiously. It doesn't transfer itself from one adult insect to the next in the environment. Instead, it's vertically transmitted through the eggs of the insect. And in order to be able to spread without being infectious, it manipulates the reproduction of the insect. And this phenomenon is known as cytoplasmic incompatibility. And what we see, if you have insects here, a mosquito with Wolbachia, the male, and a female without, we see that all of the eggs of this cross all die at a very early stage. There's a developmental defect that occurs, and I won't go into the detail of the molecular and cell biology. There's been some very interesting developments in the last year, some quite strong papers published in Nature and Nature Microbiology in the last few months, providing new insights into how this works. But the interesting thing about it is that this cross results in embryonic death of the host insect. But this cross, the reciprocal, where the female has Wolbachia and the male doesn't, the eggs all have Wolbachia because it's maternally transmitted through the eggs, but all of those eggs will hatch into mosquitoes that will carry Wolbachia. And similarly, if both parents have Wolbachia. And so using this mechanism in almost a spiteful way, Wolbachia is able to increase in frequency within the host population, and very dramatically. I mentioned to you that Wolbachia has been known to interfere with a number of human pathogens. This gives you some lists of the pathogens that have been examined and where we have scientific papers in the literature. So all dengue viruses, all serotypes of dengue have been shown to be blocked by Wolbachia in the mosquito, as has yellow fever, West Nile virus, chikungunya, Zika, amazingly filarial nematodes as well. We're seeing reductions, and also malaria parasites. Based on what has been published already, we can make predictions that we expect other flaviviruses like Japanese encephalitis to be blocked, other alpha viruses like Semliki-Forest virus or Venezuelan equine encephalitis virus should be blocked as well, we would predict, and also other species of Plasmodium and filarial nematodes. Do you mind if I steal your water just here? Thank you. That's okay. So let me just show you a little bit of data on this blocking, because it's a little bit complex. This is some work from our collaborator Cam Simmons' group out of Ho Chi Minh in Vietnam. And he's working in a hospital there where we're seeing a number of people coming into this hospital with quite sick with dengue fever. And in this data set, what you're seeing, each of these rows is a different person presenting at this hospital. They're all infected with this dengue 1 serotype of dengue. This is the viremia. Some of these viremias down here are extremely high. These are very sick people. And what has been done in this study is that blood has been drawn off these people and then fed to mosquitoes that are either just normal wild type Aedes aegypti from Ho Chi Minh City, or the same mosquito, the same genetic background with Wolbachia, the wMel strain of Wolbachia added. And that blood has been fed to the mosquitoes. The mosquitoes have been incubated for either 10 or 14 days, that you can see here. And then the saliva has been taken from those mosquitoes. And without showing you pictures, I'll just say to you that's a very technically arduous thing to do, a difficult thing to do, to take the saliva from the mosquito. And then when they get the saliva, they then inject it into other mosquitoes, into the body of the mosquito. And these mosquitoes don't have Wolbachia, as a way to amplify the virus, because you get very small amounts. And so then these columns are showing you the amount or the numbers of individuals, mosquitoes, that then are positive for dengue, that are a growing virus, suggesting that the saliva has been infectious. And just to make the data easier to understand, red is bad, and that means there's a lot of dengue in the virus, in the saliva of the mosquito, and green is good, meaning that there's very little. And I think just by looking at this, you can see WT is wild type. At day 10, you can see more orange and red in the absence of Wolbachia than there when Wolbachia is present. and day 14 as well. So this is just for, dengue has 4 serotypes, 4 major groups of viruses in dengue, and this is just dengue 1 because at this time this experiment was done, it was the dominant circulating dengue virus in Ho Chi Minh City, and so that's reflected then in the patients that come into the hospital. But there are also patients coming in with dengues 2, 3 and 4, not as many, and you can see a similar pattern here. In the presence of the Wolbachia, we're seeing much less infectious virus. But there's still infectious virus present in the saliva. It's not a complete block. And, my background is more as a laboratory scientist, and so when I look at that, I don't really know what I can say about it from the point of view of disease transmission. And so often when I get to a point where I don't know what to do, I collaborate. And so I collaborate with an infectious disease modeler at Imperial College, Neil Ferguson, and he's able to generate simulation models that can feed this raw data into and make predictions about the level of reduction in dengue disease that we should see in communities. And these simulation models have been run over a 100-year period and in different R0 settings. So R0 in infectious disease epidemiology is the number of new cases any existing case should generate. So 1.5 means that for every case of dengue, it should generate 1.5 new cases of dengue. And so typical R0s for dengue range between 1.5 and 4 in most settings. And what we can see for dengue 1 is that we get almost an immediate, complete 100% disruption in transmission in his models. And that blocking in transmission extends for around 40 to 50 years. And then he sees a small amount of transmission reoccur at the most extreme R0 setting. All of these lines are overlaid, obviously. But for dengues 2, 3 and 4, we see the blocking is a little stronger, and he sees no rebound effect out to 100 years. Looks like complete disruption of disease transmission. And so this model suggests a very large impact on the effect of that partial blocking that we're seeing. Even though the blocking is partial, we believe we have a very strong impact on disease transmission. And here's just to show you a little bit of data on Zika as well. So this was recently done by our collaborators in Brazil. What you can see here is looking at Zika copies in mosquitoes that have been fed blood meals spiked with Zika virus. And the wild-type mosquitoes here, we see a lot of Zika in the saliva. When Wolbachia is present, we see some copies that are detected by PCR, RT-PCR, in the saliva, but we see a lot with none at all. But if we take that saliva and then inject it into either wild-type mosquitoes without Wolbachia, they're all into uninfected mosquitoes, but the saliva has come from either wild-type mosquitoes or from Wolbachia-infected mosquitoes. We find that we don't get any sense of infectious virus from the Wolbachia-infected with Zika. So it looks like the blocking effect I've shown you for dengue also occurs for Zika, also occurs for chikungunya and a range of other viruses. And again, the prediction from the modelling should be a very large impact. So with that sort of basic science result, we've been very interested in how we might be able to take that into the field and see whether we can actually get some impact on disease transmission. And to think about how you would do that, it's really quite simple. You could imagine in this slide, this is time across this axis here, the x-axis. You start with a wild-type population of mosquitoes. There's a period where you release Wolbachia, not for that long, until you can get the frequency above a known unstable point. And so above that unstable point, it's predicted that Wolbachia will then spread by itself through the action of cytoplasmic incompatibility until all mosquitoes in the population, or close to all, should have Wolbachia, and they should be able to block transmission. And so in reality, we do that by either having our colleagues here in Colombia releasing adult mosquitoes with Wolbachia, here in Brazil, a bucket with that eggs and water are put into to release mosquitoes, and here in Australia, we're working with school children as well to release mosquitoes containing Wolbachia. So currently, these are the sites where we are undertaking work, our project sites with Wolbachia mosquitoes, where they're being released. We've just added a new site recently in southern India, where we haven't started yet releasing mosquitoes, but in all these other locations we have. And I'd just like in the next few slides to show you some of the key results that are coming out of these studies. The first one is from Cairns in Northern Australia, where we did our first releases of Wolbachia mosquitoes in 2 isolated communities known as Yorkeys Knob and Gordon Vale. We released Wolbachia mosquitoes in 2011. We did 10 weeks of releases, and during that period we saw the Wolbachia, these lines are measuring the Wolbachia frequency in captured mosquitoes from the field. We can see that the Wolbachia frequencies increase. We stopped releasing and then we're just monitoring, and we can see that Wolbachia has maintained itself in the population at frequencies around 90% or above ever since. And there's no sign that that's suggesting that that won't continue. And we expect that these mosquitoes, when we pull them in from the field and then challenge them with viruses in the lab, they show that they have a greatly reduced ability to transmit the virus. In another city in Northern Australia, in Townsville, so just let me go back to the conclusion of this slide, why I put it up. You know, what I think is so significant about this is that the intervention is self-sustaining. So if you develop a vaccine, for example, that needs to be given to babies or young children, it has to be applied every year as new children enter the population. If you are going to try and control the disease with an insecticide, you have to spray chemical every year. And one of the big problems in developing countries is being able to maintain public health interventions because of their cost over extended periods of time. This is an ideal public health intervention, if you like, because it's front-loaded the cost and then it maintains itself. And it's much easier to be able to generate donor funding or funding to be able to run a campaign, if you like, to put in place an intervention if it doesn't need to be sustained. So a very important feature that separates this intervention from nearly all other interventions in this space. I'd like to say a little bit about another city in Northern Australia where we've been implementing the release of Wolbachia mosquitoes. Here, this is our first whole city deployment. It's a very small city though. This is just a map of the city of Townsville, you can see on the right. We did it in 3 phases. The dark green is phase 1, and then this is phase 2, and this is phase 3. These 3 phases covered close to 90 square kilometres, so a very large area of territory was covered, but relatively small number of people because the population density is very low in Northern Australia, so around 90,000 people. 87 square kilometres over Paris would be a much larger number of people that we would capture under that umbrella. And in this case, we were doing releases through eggs, where we would take here what looks like, if you're familiar in the US and maybe here in France as well, Chinese food takeaway containers that you would have your food inside. And these essentially are just waxed paper where we put some holes in them, some instructions for people to add water, some fish food and some mosquito eggs, which we give them. They put them into the backyard of where they live. The mosquitoes grow and then release themselves into the environment. And we've been doing that successfully over the whole city of Townsville, so that we have high coverage now of Wolbachia across that city. Now, this work hasn't been done in a clinical trial setting. We've been wanting to learn from this work around more entomology endpoints about how best to deploy. But we can still get some observational data on dengue from this. And this shows you dengue in Townsville for the last 15 years. So the point here that's critical to appreciate is that dengue in Townsville is not endemic, and so it comes in, is introduced each year from travellers. So Australians go up to Bali and sit on the beach and drink beer for 2 weeks, and then they get on the plane, and as they're arriving back to Townsville, they realise their headache is not just from the beer, and that they then get sicker and sicker until they realise they have dengue, and then the local mosquitoes pick up that dengue and start transmitting it. And so these lines here are showing you imported cases. These are people that went to Bali or Thailand or somewhere else and came back sick with dengue. You can see that those numbers are increasing in frequency. Here is where we released Wolbachia and started our release. And this is the local transmission that has kicked off once these imported cases have come in. And what you can see here, that over a 12-month period and every year in the last 15 years there's been some local transmission. We've not seen any now for 3 dengue seasons. This slide's a little out of date. And showing that we've only seen 3 cases which we think could be attributed to local transmission, and those 3 cases occurring outside areas we've released Wolbachia. So this is, while it's observational data, it's not experimental clinical trial data, it's still showing supportive of the idea of very large impact. And just to say something quickly around cost here, this is the first stage of the Townsville deployment, and I just focus on this figure. We did that with a team of 12 people over 12 months. It cost us around $16 per person to do the deployment. $16 a person sounds like a lot of money when you think about other countries. But if we were to take this area of land, that 43 square kilometres, and drop it over a major urban city in say, Indonesia, then just because we would have so many more people living in that 43 square kilometres, the cost per person comes down to around $2.50 per person. And so our goal over the next few years is to see whether we can bring this cost down to less than $1 per person for implementation. The other thing to say about Townsville is that we've also been working directly with the community. And so we've developed school programs where we're working with school children, 5 to 12-year-olds, which we call our Wolbachia Warriors. And they are out there in a science program actually doing releases of the mosquitoes themselves around their homes. It's a fantastic way for us to engage their parents and the adults in the community, and has been very successful. And we've now expanded that in 2016 to more than 1,000 Wolbachia Warriors. At the same time, we're doing direct deployment through citizens within the community, and where people are coming and picking up these kits and doing their own deployments. This is just a shift to another part of the world in Indonesia. Here we're working in the city of Yogyakarta, and here we've been working in some pilot sites, again showing you dengue cases in these 2 sites here on the outskirts of the city, small sites, but showing, again, similar story. Once Wolbachia deploys, dengue cases stop, or transmission stops. And at the moment, we're doing a large randomised controlled trial in the city of Yogyakarta for gold standard evidence of impact, and that'll take around 2 to 3 years to finish at the moment. I can see this light looking at me out of the corner of my eye, and so I think I might just, a couple of slides I might skip over just quickly. This is just to say that the World Health Organization has, in the light of the Zika emergency, has been recommending the deployment of this method that we're developing for countries to examine doing pilot deployments at scale. So not experimental studies, but actual deployments. And we've started doing 2 large deployments in South America. So one in Colombia, in the city of Medellin. We are currently deploying, this is the city here in a valley. We're currently deploying over 2 million people. This green area is where we had first deployed, and now the plan over the next 18 months to 2 years will be to deploy over the whole city. And at the same time as we're doing that, we're also doing a deployment over 2.5 million people in a city of Rio de Janeiro and its twin city of Niterói. These are our pilot sites where we first started in green, and then this is the planned rollout that's underway at the moment. And we're expecting these will be deployed over the next 2 years. And so at the moment, we have deployed in the 5 countries we're working over nearly 900,000 people. And over the next 2 years, we will move that up to around 6 million people. And from those studies, we should be able to get a good understanding of impact. And so we're wanting to accumulate a global evidence picture on impact, laboratory data, which I showed you, showing reductions in dengue in mosquitoes that are working in the lab, modelling that supports that, showing the predicted impact, observational data, which I showed you in Northern Australia, before and after, if you like, showing that we're seeing no virus transmission. We have randomised trials currently underway in Indonesia and hopefully soon to start in Vietnam, which will run until the first one will complete in 2018. And then we have these large pilot deployments that we're doing in South America. And across all of that data, over the next 2 years, we think we will have a very good sense of impact. So just really quickly to summarize, because I can see that light has now turned to red, is successful pilots have now been undertaken in 5 countries. The work we're doing is community-led, it's sustainable, and we think very cost-effective. We have randomized trials commencing in 2 sites. The World Health Organization is recommending pilot deployments. Evidence should be considered as a global efficacy portfolio. And here, just to finish, I would like to acknowledge some of our key scientific collaborators that we're working with, but also to say that we have a team of roughly 400 people now working on this project in different parts of the world, and also to acknowledge some of our major funders. Thank you.
Thank you. Let's have questions. Antoine Danchin first.
Well, a straightforward question. If Wolbachia is spread so easily and sustainably, how come that the wild type are not already infected?
Yeah, so we know that 60% of insect species are infected. Why not 100%? Yeah. And so it's a good question and one that we don't really have an answer for, but I'll give you an answer anyway. And so, so I think this would be what most people think that are in the field, is that what we are seeing with Wolbachia is almost like a global pandemic of Wolbachia spreading into insect species. And at this point in time, we have maybe 50 to 60% of species infected. If we could teleport 10 million years into the future, that figure might be closer to 80%. If we could teleport another 30 million years into the future, maybe that point would be at 90%. We don't think it will get to 100% because we think species are losing Wolbachia as well. And so there's some acquisition and loss going on.
What is the trade-off? Is there an advantage not to have?
Yeah, so Wolbachia puts a fitness cost on the insect, but because of the cytoplasmic incompatibility, it can still drive itself into the insect population. But if for some reason the host is able to limit maternal transmission, or we know, just like in the coral story this morning, we know in extremely high temperatures that insects will lose their Wolbachia symbionts, that there can be situations where you may see loss. Yeah. And so Wolbachia is really a parasite that's really entering into insect populations for its own purpose. It's not really providing any benefit to the insects, although in some situations maybe this virus blocking could be a benefit.
Yeah, thank you very much, Scott, for this wonderful talk. I was just wondering, you have been using only wMel, Wolbachia. So what is the situation for the other Wolbachia? For instance, Wolbachia from mosquito?
Yeah, so we currently, most of our field studies are running with wMel. There has been a paper published recently showing that wMel may be sensitive to extremely high temperatures and could drop out. We haven't seen that in our major study sites. Maybe in one location we may have some evidence for that occurring, but it does not seem to be a general issue for us. But still, in our product development team, we have developed 6 other Wolbachia mosquito combinations, including the Al-B strain, which we know is very, very thermally tolerant. And in the next year or so, we plan to do some side-by-side comparison experiments of wMel
you didn't do it
and Al-B.
yet,
Not yet, no.
Okay.
But it's planned.
First, yes.
Yes, thank you very much. I would like to know whether you have observed mutations in the Dengue or West Nile or Chikungunya virus during these 5 years. Have you observed any problem?
No, the problem is that we're not able to get the viruses to be, because there's no transmission going on. So to look for escape variants or to look from the field has not been possible. But as we go into bigger deployments, we should be able to see whether, through deep sequencing, whether we're seeing shifts in the genomic diversity of viruses circulating in the region. We have done a number of laboratory evolution experiments to see whether we can force dengue through sequentially and select for resistance, if you like, so viruses that can escape. We have not been able to do that. I think part of what is of benefit to us is that we think the mechanism of blocking is through a number of different approaches. So there's an innate immune piece where the Wolbachia is upregulating the innate immune system of the mosquito, and we also think it's competing for key fatty acids with the virus, like cholesterol. And we think these two mechanisms need to be overcome simultaneously. And so we think, and at least to date, the work we've done suggests that resistance doesn't look like it will emerge quickly. And certainly we've seen no evidence in 5 years in the field that there is any reduction in the ability of the mosquitoes to block. So some of these questions we cannot do in the laboratory. We have to take the risk, do the field work, and then monitor and see whether we see. We should expect, as in all biological control work, that at some point we will see resistance emerge. I'm not, and the evolutionary biologists seem not so concerned about the virus becoming more virulent. Certainly, very similar parallels would be vaccination with human vaccines that are not 100% efficacious. Very well-known one would be Sanofi Pasteur's vaccine for dengue. Or you can think of any other number of others. And in those vaccine scenarios, we haven't measured any sense of increased virulence in viruses when populations of people are vaccinated. So I don't think, and the evolutionary biologists don't think that's a problem. I'm more concerned about the strength of blocking might get weaker over time. Just as myxoma virus doesn't kill European rabbits as well in Australia anymore. But if we could get 40 or 50 years of blocking, that would be enough for me, yeah.
Pascal Cossard first.
Okay, Scott, I think you know that there are other techniques which are used. Yes. And I would like to have your comments on, for example, spreading sterilized mosquitoes and things like that.
So there are some other great technologies out there. The one that is fairly well known is genetically modified mosquitoes. For example, Oxitec's work of being able to find really cool ways to be able to crash mosquito populations. The problem has been for Oxitec, and continues to be, that the general population is not very happy with the idea of having genetically modified mosquitoes released. And so there's a problem around regulation, and there's a problem around community acceptance. And so the best technology in the world, if the community won't accept it, doesn't go very far. And so I think that's why it has been a problem for them. There's been a lot of talk around CRISPR-Cas9 and the potential it has for gene drive systems that could eradicate. But again, we're seeing major issues emerging with resistance to those approaches, and papers being published around that. And then I'm just flicking back through my slides because there are some groups using Wolbachia in a different way. We are wanting to do this and release both males and females, and so convert the population to all have Wolbachia. There are other groups that are just releasing the males and trying to use that as a way to suppress. I think that's a fine approach. It's just that all of those approaches are being done at a very small scale. Hectares, not square kilometers. And the cost, if just spend 1 day in Rio de Janeiro driving around and see the enormity of the problem and what would be required to be able to mass release male mosquitoes over that entire area. And it's just the cost of that in these countries, I think, is not going to be sustainable. But obviously I'm anchored, and I have a strong belief in the technology that we're pushing. So I'd also like to say, though, that we're not for profit and we don't have any commercial interest with it.
Karina, Xavier.
Hey, great talk. I had a kind of related question. So how many mosquitoes do you need to release per square kilometer to see an effect?
Yeah.
Did you assess this? And does this differ between
So it depends a lot on how many mosquitoes are there to start with. But typically our release rates have been successfully done with releases. Our first ones in Australia were done where we released 10 mosquitoes, of which 5 were female, every fourth house. Every house, sorry. So we would do 50 mosquitoes every 5 houses. And so 10 mosquitoes per house per week for 10 weeks. In other places, we've had to do it for a longer period of time. When we talk to the community about it, most community members don't feel any increased biting pressure at those release rates. You have to appreciate too that there are many mosquitoes in the environment, of which Aedes aegypti is just one, and usually it's at a quite low density in the environment compared to other nuisance spiders like Culex.
Thanks.
Christian Dumas down there. Yes.
in front of your experiment, did you observe some complaint from ecologist group?
You know, we've spent a lot of time talking to different stakeholders, including environmental non-government organisations, and we have had no opposition really when we spend the time to talk to people and listen to what they have to say. We've done experimental work to look at concerns around horizontal transmission and potential adverse consequences. I think most of the environmental NGOs see us as preferable to GM methods that are being proposed at the moment, or the widespread use of insecticides in the environment. And so if anything, we are being held up as the green alternative for current mosquito control technologies. Yeah.
Eric Orsena would like to make a comment.
Thank you, Scott. So far in my book, Wolbachia has only five lines. And now I think Wolbachia will have more than 8 pages. Thank you.
That's good. Thank you. I'd love a copy.
Yes, exactly. This is the oh, would you like to say something?
So just curious in terms of you increase the frequency of Wolbachia in the populations, but what's happening in terms of numbers of mosquitoes?
So Aedes aegypti is really hard to do population biology on in the field because of it's very focal. You have these volcanoes almost of aegypti here for a few months and then they disappear and then they're over here. And so to get reliable population estimates is quite difficult to do unless you do very large experiments. And so we're wanting to try and do that at the moment. We certainly, as part of our regulatory approval permits, we've had to measure mosquito populations to make sure they don't get bigger. And certainly our data doesn't show that populations are getting bigger. If anything, we think they're slightly smaller, which is what the modelling predicts they should be because of the fitness cost. But that difference looks fairly small. And so we need a large experiment to have the statistical power to be able to really measure it. Yeah.
All right. Thank you again.
Thank you.